Hello everyone, I'm excited to be here. I'm Anu, pursuing PhD at Georgia Tech with Saurabh. Today I'll present our InSTanT toolkit for intracellular spatial transcriptomic analysis. So recently we have seen large amount of data generated at single molecule resolution from technologies like Murfish and Sheepfish. In the left panel, each dot corresponds to a cell, and in the right panel, you have a zoomed in version of a cell where each dot corresponds to a transcript. In this project, we asked a simple question: what can such data reveal about subcellular organization? In our quest to understand subcellular organization inside cell, we define a new statistical phenomena which we call as decolocalization. Here D stands for distance D, which is 4 microns in our experiments. Decolocalization of a gene pair is a statistical phenomena which aims to detect gene pairs that tend to occur within distance D of each other. For example, InSTanT Toolkit detected a pair PRKDC and FASN as decolocalized, and you can see in 4 sample cells the transcripts of corresponding genes are nearby each other. Like other statistical phenomena, such as differential expression of a gene or co-expression of a gene pair, decolocalization may serve as a starting point for discovery of underlying biological relationships, such as RNA-RNA interactions with regulatory functions. So now I will present our statistical tests to detect decolocalized pairs. First, we perform a proximal pair test, which aims to find if a gene pair tend to co-localize in one particular cell. Next, we have a conditional Poisson binomial test, which aims to find if a gene pair tend to co-localize across many cells. Both of these statistical tests were carefully designed to remove any bias or unwanted error, which I will describe in next few slides. So first is proximal pair test. You have a cell here. Each dot corresponds to a transcript. We want to find if gene 1 and gene 2 are co-localized. You can see there are 3 instances of gene 1, red in color, 2 instances of gene 2, and total 2 pairs of gene 1 and gene 2 are co-localized. So there are 2 co-localized pairs. Total possible pair is 6. So we ask simple question: is 2 out of 6 significant? So we do a binomial test where null probability is obtained by creating histogram of all pairwise distances between all the transcripts and reporting the fraction of gene pairs that is below distance t. Once we have results from PP test for one cell, we will use that for which will be input to our CPV test. So here you have many cells. For each cell, you do PP test. A PP test will give you a co-localization matrix, where for gene I and gene J, this is a binary matrix showing if this pair was co-localized in this corresponding cells. For example, in this cell, this pair was co-localized, so we have 1. We can also get null probability of obtaining a pair being co-localized here. This is obtained by calculating total number of co-localized pairs in this cell, which is 3, divided by total number of possible pairs, which is n choose 2. We can similarly perform PP test for each cell, 1s and 0, and we'll have different probabilities for each cell. Note that we have different probabilities because some cells may have many co-localization, some other cell may not have co-localization. And we would like to ask if the number of co-localized cells, is it enough? So this distribution is a variant of binomial distribution. Just that you have point toss. The difference is different coin toss has different probability, and it's called Poisson binomial in literature. So finally, you use that Poisson binomial to get a p-value for each gene pair. Now, a major assumption in our previous formulation was that all of the genes are equally likely to be co-localized with each other. When we generated the results using that formulation, this yellow in color, you can see as we had a higher number of co-localized pairs, we also saw that at least one of the genes was an abundant gene. It was because that abundant gene is highly expressed. So we wanted to remove this bias. So previously we had formula, null probability formulation is this, PIJC is basically probability of I and J gene pair in cell C equal to number of co-localized pair in that cell divided by total possible pairs. Now we'll have a new formulation. The numerator is still same, but we have an updated weights, which I represent as Wij. Previously, Wij was same for all the gene pairs, which was 1 upon n chose 2. Now it will somehow take care into account if a gene is highly expressed or highly co-localized. Once we use this new formulation, we can see as we have higher number of co-localized pairs, the probability that any gene, decolocalized gene pair has an abundant gene greatly reduces. For biological interpretation, we also annotate each gene pair with the most common region it co-localizes in. So we have nucleus, perinucleus, cytoplasmic, and cell periphery. We observed high number of co-localized pairs in perinucleus region and nucleus region, but we also observed around 20 co-localized pairs for cytoplasmic and cell periphery region. Below, you can see an example of co-localization in each region, nucleus, perinucleus, cytoplasmic, and cell periphery. Now we needed a scheme to assess our method. So we developed a randomization scheme. To do so, we randomize a gene pairs label for each transcripts in a cell. This allows us to have same spatial distribution of transcripts, but any gene-gene relationship is removed. We run our PP test followed by CPV test. Any pair that is detected as decolocalized is clearly a false positive. Here you can see that for D equal to 4 micron, we are able to detect 400 co-localized pairs with a false positive rate of 0.05. To also see if our results are reproducible, we ran our test across different technical replicates and saw around 80% overlap. Now you may be curious, what are the functional consequences of decolocalization? For this, we define a gene pair to be functionally related if they both are annotated with same geoterm or are in the same pathway. For example, we found 461 gene pairs in which both of the gene pairs were annotated with the same molecular function geoterms. And we also found 403 decolocalized pairs. We saw that 67 pairs were overlapped, which was significant at the p-value of 7.6 e-17. On further analysis, we saw that all these 67 pairs, common pairs, were annotated with term RNA binding. We also saw significant enrichment with other geoterms as well as pathways. Another possibility of decolocalization can be due to RNA-RNA interaction. We found few genes whose decolocalization may be explained by RNA-RNA interaction. Here you see a result for one gene, USB9X. We calculated RRI score from RNA-Plex software, and all the red cross denotes the decolocalized pair in which at least one of the gene is USB9X. A score above 35 means they are interacting. You can see these red cross, they are higher number compared to its background distribution. We also found one really interesting biological phenomena. So we did the same analysis only on nuclei of all cells. And we found that SRRM2 and MALAT1 co-localize together. We came to know about a study that says that a nuclear consists of nuclear speckles where splicing happens, and it's known that MALAT1 co-localizes to nuclear speckles. It's also known that SRRM2 protein is a core structural component of nuclear speckles. Decolocalization of SRRM2 RNA and MALAT1 may shape our understanding of how splicing happens in nuclear speckles. So now I will go to give a big overview of whole InSTanT full functionality. So we have a tissue, we do PP test and CPV test, and this gives us a decolocalization map. I showed results till this. Now we can use this decolocalization map for further downstream analysis. I'll show results for three of these kind of analysis: cell type specificity, spatial modulation, and gene module discovery. To enable this analysis, I'll move to another data set which has different cell types. This is mouse brain dataset, and it has 9 cell types. So our first analysis does basically categorizes a decolocalized pair into 3 categories based on cell type specificity. For example, a pair ALDH-L1 and MLC1. This is a decolocalized pair, but it's found in almost all of the cell types. While we have another pair which is more specific to inhibitory neurons. Next, we wanted to ask if there is any correlation between intercellular and intracellular spatial information. In particular, co-localization in a cell may be influenced by its neighborhood cells. So we propose a probabilistic graphical model where the probability of co-localization in a cell is dependent on its neighborhood. And through this, our model is able to output all those gene pairs which are decolocalized as well as spatial modulated. We divided these spatially modulated decolocalized pair into 2 categories. One is again cell type specific, this one, and again this one, the first panel is not cell type specific. So you can see COL25A1 GAD1, this is the cells here, the blue dots, they have a spatial region in which they are actually co-localized. In t-SNE plot, you can see these black dots are scattered throughout all cell types. However, another gene pair, which is CD24A and MLC1, this co-localizes in a strip region here, and it's only present in one of the cell type. We also developed 2 methods of finding if a cluster of genes co-localize together. First, we'll present our first method called global co-localization clustering. It aims to find if multiple genes may co-localize together. We just do hierarchical clustering on our decolocalization map. Here you can see we have 2 modules. When we do geo-enrichment analysis of module 1, we see terms like related to RNA binding and cytoskeleton organization. And you can see a sample of 3 cells in which this module is co-localized. You can see this co-localization mainly happens in perinucleus and cytoplasmic region. We also have another method which enforces that co-localization of the whole modules occurs in the same cell. For example, we found this module consisting of 4 genes in which all of all 4 genes co-localize in the same cell. Geo-analysis showed terms like myelin sheath and cytoskeletal protein binding. And here is the sample of 2 cells where these 4 genes are co-localized. You can see this co-localization is happening near cell periphery. This dataset didn't have cell periphery or nucleus boundary. So in summary, we developed our toolkit has 2 most important tests, PP test and CPP test, which creates a decolocalization map for a data set. And then you can use this decolocalization map to do further downstream analysis to understand better. I'm happy to take any other question. Thank you.
All right. I was wondering if you have a chance to look at the, the potential regulatory mechanisms of like a co-localization. For example, looking at the UTR subcellular like localization signals are enriched for the pairs you find from this like a decolocalization, or like they are like located regulated by a similar transcription factor in their genomic coordinate.
Yeah, that's a very interesting question. In fact, this was the thought which have been going in my mind in last 1 month, but we have not been able to look up. I think once we define this statistical phenomena and after we publish, that's the next step. To have basically, you can do differential GRN and maybe focus on only those regulatory edges which is also decolocalized. Yeah, that's the next step, and I'm looking forward to that.
Very fantastic. Looking forward to it.
Hey, thanks for a great talk. So For the hypothalamus data, right? Presumably, you don't just have nucleus and cell body, but also axons and dendrites. Because at least for the neural clusters, did you look at whether or not there were co-localizing RNAs in some of those projections?
Okay, so I think the data we have is only of the soma, and so we don't have data of synapses. That's very difficult to get.
Oh. Okay, thanks.
But yeah, if we had data, we would have very interesting results on this.
Okay. Thanks. Oh, there's an online one. Do you want to read it? Or? Are all transcript pairs of the same gene pair treated? Are all transcript pairs of the same gene pair treated?
Of the gene pairs. Yeah, okay. So I think, yeah, in a cell, let me go back to our PP test. If I understood the question correctly, the question is when we try to analyze when we try to find if gene 1 and gene 2 co-localized in a cell, are all the transcript pairs considered? Yes. You see there are three gene 1, 2 gene pairs. So N is total 6. So you then ask question if 2 co-localized pairs is significant in 6. So yeah.
Okay.